Hi, I'm Evie, the Evernia prunastri, and I'm a lichen, just going about my daily— Oh no! What are these strange things attacking me? Microbiome to the rescue! But wait, what's a lichen?
The natural world is full of mutualisms, and lichens are an excellent example of this. Considered for centuries to be a symbiotic relationship between an alga and a fungus, in recent years, researchers have discovered that the lichen system is more complicated than we thought. Lichens have been shown to contain multiple species of endolichenic, i.e., living within a lichen, fungi and bacteria alike, all of which play complicated roles. Today we want to focus on the microbiome of lichens, a group of many bacteria living inside the lichen thallus. These bacteria have been shown to benefit the lichen, specifically providing defense from encroachment by inorganic elements such as arsenic, copper, and zinc, as well as fighting off fungal and pathogenic invaders. Our study looked specifically at a lichen species called Evernia prunastri. Based on our background research, we hypothesized that there might be a relationship between the diversity of the Evernia microbiome, the amount of infection present on a lichen. We looked specifically at infection by a parasitic fungus called Unguiculariopsis lataui, and the amount of nitrogen, i.e. pollution, found in its tissues. Since we know from past studies that the lichen microbiome is a powerful hero that's great at fighting off evil villains like infection and pollution, we thought that a diverse microbiome and low levels of infection would go hand in hand. A paper by Wang et al. From 2018 found that nitrogen deposition in lichens caused their fitness to decrease. We thought that increased nitrogen, then, would make lichens more susceptible to infection. Put both of these hypotheses together, and what do you get? A third one that says that higher levels of nitrogen pollution would cause the diversity of the microbiome to decrease. Let's get to testing. We and our classmates spent the entire quarter collecting data on lichens from Jasper Ridge Biological Preserve, a spot not too far from Stanford campus that has a lot of Evernia. We then counted the number of fungal ascomata, small black dots that are the reproductive structures of Unguiculariopsis, and considered that number compared to the mass of each lichen as our metric of infection. Next, we sent the little lichens off to a few labs. One of them measured the amount of nitrogen present in their tissues. The other lab sequenced the microbial DNA of each of the lichens. And what we found was interesting. Notably, the sequencing data came back with 7 different types of bacteria, all of which have been observed before in the lichen microbiome. We had Rhizobiales, Bryocella, Calobacteriaceae, Methylocella, Sphingomonas, Endobacter, and Acetobacteriaceae. Some of the bacteria are even kind of niche. They're methanotrophs, meaning they can metabolize methane. What? Unfortunately, none of the analyses of our hypotheses turned out to be statistically significant. But all this really tells us for now is that we don't have enough data yet. Only 12 lichen samples came back with usable sequencing data, which significantly limited our scope of investigation. But this is still exciting because bacterial sequencing data has never been collected as part of this class before at Jasper Ridge Biological Preserve. And just because our data wasn't significant also doesn't mean our findings are useless. We decided to do some digging into the various types of bacteria we found in these lichen samples. We learned that some of the bacteria living in our lichens are particularly sensitive to heat, namely Bryocella. Does this concept ring any bells? Some researchers, Maitri and myself included, think that lichen microbiomes could possibly give us a way to look at how climate change is affecting the lichen mini ecosystem that we talked about earlier. A paper published in 2020 by Klarenberg et al. Mentions several of the genera of bacteria found in our lichen samples from Jasper Ridge, like Endobacter, Bryocella, and Sphingomonas. The researchers found that the bacterial composition of lichens is likely to change with a warming climate, but that after long periods of warming, they can bounce back to their original bacterial makeup. Are lichen microbiomes a microscopic microcosm of climate resistance? What can we learn from them? There's no way to know for sure until we do more research. There's so many exciting avenues for future BIO46 classes and the wider scientific community to explore in the future. In the meantime, shout out to Evernia for being the cutest study system of all time. Thanks for watching, and see you in the woods.